So our final presenter this morning is Tai Tulo Mekonnen from the University of Sydney. Tai is a postdoctoral research associate, and he received his PhD in biomedical engineering from Macquarie University, where his notable contribution was the development of pioneering imaging technology called multi-channel optical coherence tomography. His current research is focusing on a high-resolution version of this to assess dynamic behaviour of dry powders, and this is what he's going to come and talk to us today. Welcome, Tai. Thank you.
So, yeah, I will be talking about our recent developments on characterizing deposition of dry powder in inhalers using an imaging technique known as optical coherence tomography, as called OCT in short. It's well known that inner walls of DPI devices are crucial in contributing impacting the aerosol retention. So characterizing the local deposition inside the DPI is normally useful to optimize inhaler designs and as well as for validation of CFD models. But with the common approach of measuring deposition inside a DPI is obviously based on the total deposition, but that doesn't fully capture the in-device dispersion and the deposition characteristics, which is normally related to which would normally characterize the powders themselves as well as the device performance. So measuring local deposition is something quite useful in that respect. However, the main challenge is this measurement of a local deposition will require the access to the inner walls of the DPIs, which is not quite feasible to do because of actually just from the geometrical perspective and also the makeup of the inhalers. The other thing is there is really not a very suitable technique to do measurements inside the DPIs themselves. So as a solution for the first problem, what our group has come up with is an optically accessible DPI device, which can be used as a testing platform for the various DPI devices. This device, which we call OA DPI, would actually simulate the number of flow conditions of typical commercial inhalers. For example, the DPI has, as you can see in here, would have 2 inlets and there is 1 outlet. It has got a swirl chamber common to some commercial inhalers like aerosolizer and osmohaler, and also has a matching resistance also. So with this characteristic, this device would actually enable inhaler performance evaluation applicable across various DPI devices. One of those characteristics that could be analyzed would be the fluidization and the particle dynamics. In this respect, our group member has recently published a paper characterizing the flow dynamics inside this DPI device using a high-speed camera, which is actually using a laser backlit to illuminate the plane of imaging here. The other characteristic which I'm going to present in here is the particle deposition inside the DPI. So yeah, when it comes to deposition measurement, most of all, you know that deposition can be measured using a different technique, but what our group have come up with is a novel approach using optical coherence tomography. OCT, just to give you a highlight, is a tomographic imaging technique, which actually is non-destructive. You simply project a beam into your sample and then scan across the sample would give you a subsurface, not only a surface microstructure, but it also provides a subsurface microstructure. But even though OCT is an established technique nowadays in the various fields of biomedical, such as ophthalmology, but the existing commercial devices cannot directly be used in this DPI imaging. So we developed our own in-house customized OCT system that would enable measuring the intact deposition inside the DPI itself. We have made a number of technical and data processing changes, which I'm not going to detail it in here. But simply put, OCT is just you project a beam onto a sample, and then we scan by using some Galvo technique. So it provides, for example, if you point at a single point in the sample, we get one depth profile, and by repeating the same, we could get multiple depth profiles, which can be reconstructed into one cross-sectional image, and then we finally get a 3D image if you repeat this cross-sectional imaging. So we demonstrated this technique using an airway model previously, which is made of a silica. This was actually reconstructed from a CT scan. It's so a realistic airway model. So we measured the deposition at different positions, or local depositions. We were able to reconstruct the deposition profile at different positions along the airway for actually the first time. So we are translating this technique into a DPI now, as I already mentioned, projecting the beam into the inner walls of the DPI itself, and then we could actually construct a 3D image that actually provides the volumetric view of a deposition at a particular position. Then we can do a lot of post-processing to characterize the deposition characteristics. So for this, we just set up a simple flow system that actually is to just simulate the inspiration flow, and the DPI is connected to it, and then OCT would actually scan at a region of interest. For example, in here, this rectangular region, simply selected to analyze assess the feasibility of this method. So for this particular study, we actually used in-house formulation, particularly 5 formulations with one baseline lactose, so SVT, where the API was actually sylbutyl sulfate SS, as abbreviated in here. So different proportions were actually selected based on some experimental design parameters, and also to get a wide range of SOX number for the fine lactose and the coarse lactose, so that we could be using the data for CFD simulations. Yeah, so just to give you how the data is being interpreted. For example, if you take a measurement at this position, and then the XY actually represents the in-plane, or the surface of the inner surface of the DPI, and then the Z is into the surface. So we are able to capture the subsurface information in there. So 3D views of the images can be reconstructed. And then thickness profiles can be calculated from the individual frames inside the image like this. Yeah, so comparing the formulations, what we observed was instead of actually a uniform distribution of deposition, it was found out that the depositions happen to be clusters of blobs which is spatially scattered over the surface of a DPI. And these deposition profiles were different for the 5 formulations compared to SCV10. And we further tried to quantify the thickness of the depositions for the different formulations. For that matter, we just grouped the formulations into 2 to assess the effect of the API and the fine lactose. First is for the different proportions of the API. Second, the graph represents this for different proportions of the fine lactose, but fixed amount of, as you can see in the table, fixed amount of the API. So change in SS fraction appears to influence the peak thickness distribution, but the effect was actually not significant. But the change in the fine lactose actually we found it to correlate much with the increase in deposition thickness. Yeah, so in summary, obviously OCT showed a very promising approach to quantify depositions, not only in the in vitro airways, or it could also be done inside the optically accessible DPIs that would be used to access the deposition characteristic. And local depositions were found to form some form of clusters. Yeah, so as an outlook, this technique, not only can we actually measure the deposition in microstructure, but we also found that the deposition can be measured in real time, like how the deposition can be accumulated over time, or does the particles normally stick to once they deposit, they stick to the surface or re-enter the flow. That means we can also measure the deposition rate as well as the reentrantment rate. So we can also do some spectroscopic analysis to characterize the blends. Yeah, so finally, I would I would like to thank our group as well as our funders, Australian Research Council and the FDA. Yeah, so that's all. Thank you so much.
Thank you, Ty. A really interesting presentation. Do we have any questions from the floor? I've got a couple while we're waiting. I guess the first one is, how did you choose the material that you used for the design of the aerosolization? And what do you think the impact of material change or the surface properties of the material might have on your deposition profile?
You mean the material for the DPI
Yeah.
itself? We chose the acrylic for now, just because we found it to be optically accessible.
Yeah. Okay.
And we actually made sure that the surface is really smooth so that it doesn't affect the depositions. But materials that would actually emulate the actual DPI devices would be something which we are actually aiming to do with in the future. Yeah.
Thank you. Any questions? Anyone? Put your hand up. Okay. In that case, I've got one other question, if you don't mind. The region of interest that I think you showed looked like it was quite close to the powder bed, I think, from what I saw in your diagram. And I was just curious to understand whether you'd looked at different regions of interest and what the general, any variance that you may have seen in the deposition profile across the actual swirl chamber.
Yeah, so initially we tried at different positions in the swirl chamber. We found that at most of the positions, the deposition profiles doesn't change much, but the one thing was that at the entrance, it was found to be very much consistent between different measurements. That's why we actually chose that region. Otherwise, measurement can be done anywhere inside the swirl chamber. And we are actually planning to measure the whole region of the swirl chambers by actually implementing a wide field of view of imaging using this OCT.
Yeah, it'd be interesting to see if you do see the sensitivity of that variability to different formulations, because obviously it will be impacted by the dispersibility of the powder. Yeah. Okay. No, thank you. And we've got, I think, 2 minutes left on the time. Has anyone got any final questions for Taye? If not, thank you very much. Great presentation.
Thank you.
Have a good time.